Okay, hello everyone and welcome. My name is Joshua Stein and I'm project manager at the International Wheat Genome Sequencing Consortium. And it is my pleasure to serve as moderator for today's webinar. So welcome everyone. Uh, while people are still joining the meeting, I just want to mention, so we'll be joined in a few minutes by Yves de Smet of the VIB-UGent Center for Plant Systems Biology in Belgium. So we're just gonna wait a few more minutes because there's still participants joining. Okay, so before we begin with Eve's presentation, I just want to quickly give an introduction to the IWGSC and a quick overview. So the IWGSC is a international nonprofit with 3,500 members from 964 institutes and companies coming from 78 countries. And we currently have 7 sponsors that help fund events like this. And so I want to give them acknowledgement as shown here: Arbor, BASF, Smet, Florimond Dupre, INRAE, Kansas Wheat, and RAGT. So thank you, sponsors. So as many of you know, the IWGSC vision is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And with that in mind, I want to remind people that we have initiated the Wheat Diversity Project since last January. So we're about 10 months into this project, and the project goals are to preserve, discover, and mobilize genetic resources by sequencing landrace genomes that encompass the worldwide diversity of bred wheat. And so our aims are listed here. They include assembly of 12 bred wheat landrace accessions, as well as Chinese spring. And then we additionally have plans to generate transcriptome data to perform gene annotations. And I want to point out that we will be producing a new version of the Chinese Spring RefSeq, and we hope to maintain the annotations from the existing version. And then finally, all of these data we plan to package as a practical haplotype graph in order to do inquiries. And I also want to thank the funders, NSF, and the European Union predominantly for funding this project. I also want to remind people next in just a little over a month, December 4th, we'll have our next webinar. And so we'll be joined by Tally Wright of NIAB of the UK, and he'll be speaking about applying quantitative genetics to uncover the potential of diversity-enriched wheat. So please join us then, and there's a link here to register for that. I also want to remind people of the upcoming Plant and Animal Genome Conference in San Diego that's occurring in January. As usual, we'll have the IWGSC main workshop on Saturday. And on Tuesday, we'll have the IWGSC From Structural to Functional Wheat Genomics workshop, which I'll be chairing. I believe we'll have really great speakers. So I think this would be a great opportunity for people to attend. Also on Saturday, we will be celebrating the 20th anniversary of the IWGSC since its founding in 2005. Okay, so before we begin, I just wanted to quickly cover the webinar dashboard. You can ask questions throughout the presentation, but the questions will be responded at the end. You can use this button on the dashboard to ask questions, and then the chat panel here will be used to communicate messages and handouts and things from the organizer of the webinar. Finally, I'm very pleased to introduce Yves de Smet from the VIB-UGent Center for Plant Systems Biology in Belgium. And the title of his talk is Comparative Wheat Phosphoproteome Profiling Pinpoints High Temperature Associated Breeding Markers. So let's welcome Yves. Thank you very much. Yves, are you ready to go?
Can you hear me?
Now we hear you.
Okay, brilliant. Yeah, and the screen is on. Perfect. So yeah, thanks for the invitation. So I'll be talking a little bit about what we have been doing in the context of wheat phosphoproteome profiling, how this might help in breeding. And then I'll also go a little bit into some mechanistic details with respect to particular environmental, physiological, developmental responses associated with elevated temperature. So just to kind of put this a little bit into context, obviously abiotic stress impacts wheat yield, and there's kind of various yield losses due to abiotic factors. And very specifically, what we have been focusing on is high temperature. And high temperature is predicted to lead to a dramatic decrease in yields, up to 6–7% per degree increase above the optimal growth temperature. So that's kind of the context that we have been working on. In addition to these yield losses, we have primarily focused on early growth and development. So if we put young wheat seedlings and we grow these at different temperatures, which are either slightly below or slightly above their optimal growth temperature, it's pretty apparent that if you put them at the lowest and the highest temperature, their kind of growth and development is perturbed or at least delayed. If you then have temperatures that are optimal or slightly above optimal, you tend to have maximal elongation of these seedlings. So if we take a more kind of quantitative look at that and indeed try to phenotype these young seedlings at a given stage while they have been growing at these indicated temperatures here, then we immediately see that there is, specifically focusing here on the third leaf, there is this kind of growth response along this temperature gradient, which kind of nicely reflects the plasticity of these wheat seedlings along this temperature gradient, and that there is an optimum where, for the varieties that we've just tested here, there's an optimum that lies more or less in between 24–29 degrees, where you get maximum growth. Now, of course, this also very much depends on the variety. So we've tested varieties that have, based on other physiological yield and developmental parameters, been indicated as being either tolerant, medium tolerant, or sensitive to elevated temperatures. And then we see that indeed, depending on these predicted responses, the maximum growth varies a little bit. In addition, it's also clear from these images here that the response, and at least the uniformity of the response, very much depends on which leaf we are looking at. So we tend to see a fairly uniform response from the third leaf onwards. We do the same kind of analysis in the first leaf, and this is improving a little bit in the second leaf. The growth responses are a little bit all over the place. So we kind of tried to group these responses and cluster them along this temperature gradient. And then indeed, based on our clustering profile, we do see that we reveal the tolerant and medium tolerant as one group and the sensitive ones as another group. And we've primarily focused on these two varieties here in the context of the sensitive ones, which are these Caducean Reeves varieties. And then we've explored cadenza, a little bit Chinese spring, and to some extent Halbert as well in subsequent analysis. Now, what we have been focusing on is very specifically how the environment changes the protein level. So we've been looking at proteins, proteomes, by extension as a collection of these proteomes, and not necessarily at DNA level genome or RNA level transcriptome. And very specifically, we have been looking at post-translational modifications. Because if you look at a single protein, and this is of course, if you take all these proteins together, bringing up the proteome, all of these individual proteins can undergo a number of post-translational modifications, which are either reversible or irreversible, and these impact the functionality, the activity, and so on of these particular proteins, which means that one gene is not necessarily encoding one particular protein, but is encoding a diversity of proteins based on these modifications. And if you then look at single proteins and you kind of look at these into a complex proteome, you see this kind of multitude of modifications coming together on individual proteins. So trying to understand what these modifications do, what they achieve, but also trying to just capture these modifications on a proteome-wide level is not trivial. So we have been predominantly looking at phosphorylation, reversible modification on these serine, threonine, tyrosine residues that are mediated by kinases and phosphatases. We also looked a little bit at structural changes, but I won't go into that today. We have been looking at these phosphorylation events under temperature conditions and a little bit under drought conditions, because obviously high temperature and drought are intimately connected. And we have been largely looking at this in the context of shoot growth, a little bit at fertility, and mainly in three plant species, which was Arabidopsis as our model dicot plant, a little bit of our analysis through an FFAR-funded project together with Anna Locke and Ross Sozani in soybean. But what I will talk to you about today is predominantly the work that we have been doing in wheat. So why phosphorylation? Obviously because it impacts a large number of protein-related functions, activity, the localization of the protein, crosstalk with other proteins, conformational changes, and that's why we also have been looking a little bit at structural changing, turnover, and binding with other partners. So we spent well, basically a little bit more than a decade ago, when we started this work, we spent some time on trying to indeed come up with a workflow to capture and enrich these phosphorylated proteins, and then look for proteins that are phosphorylated and changes that are really due to an alteration in kinase and phosphatase activity, and not necessarily through a change in protein abundance. So we've kind of developed this mass spectrometry-based workflow to capture the truly significant deregulated phosphosites. And all of the information that we have been collecting can be looked at in this plant PTM viewer, which we have been developing together with a number of colleagues within the VIB, which is not only reflecting changes in protein phosphorylation, but also numerous other post-translational modifications. And you can there look at your favorite protein and check for what kind of modifications under which kind of condition, experimental context, or mutant background these modifications occur. So feel free to take a look. Now, the approach that we have been following in the lab, in the group, is essentially that we do phosphoproteome analysis in wheat to identify relevant candidates, potentially relevant candidates. We then try to validate these in wheat initially by looking at loss of function lines for these particular proteins. And then we try to gain a little bit of a deeper understanding, mode of action studies by mainly using Arabidopsis. And to the extent possible, we try to then also go back to wheat. So I'll try to outline some of these examples throughout my presentation on how we come to then deeper insight. Okay. So coming back to the experimental setup, which I mentioned before, where we have been growing our seedlings at different temperatures, kind of slightly lower, slightly higher temperatures, which kind of gives a growth response. So now we kind of were wondering to what extent is this growth response and this kind of typical Gauss curve response, to what extent might we also see that in the molecular biochemical response. So for that, we exposed seedlings to the same kind of temperatures, but only for 60 minutes. And then we extracted the proteins from the leaves, and part of these proteins will be phosphorylated. We then digest these proteins into individual fragments, so we kind of break up this protein into small fragments, which we can then analyze using the mass spectrometer. And in addition, to really specifically focus on the phosphorylated residues, we do an enrichment for only those fragments where we can see phosphorylation. And then we subject these to LC-MS-MS to really identify the amount and the identity of these specific phosphorylated peptides. So when we do that for our temperature gradient, we see that there are indeed a number of peptides, and each dot here represents an individual peptide that are not obviously different between the different temperatures, but we also see clouds coming up in this UMAP plot that tend to be associated with one particular temperature, and one particular temperature that has a particular growth response in these seedlings. So we're kind of reasoning that the growth response that we find along this gradient is also encoded in a way in the kinase and phosphatase activity, and that key differential phosphorylation of a number of proteins is driving these responses at these specific temperatures. Now, when we look at these data from a different kind of point of view, we start to see a picture where we basically identify three distinct clusters. So on the one hand, and this is also reflected here in this correlation plot, on the one hand, the lower temperatures, 14, 19 degrees, tend to group together. The intermediate temperatures, the more optimal temperatures, tend to group together, 24 and 29. And then we have the more slightly elevated temperature that also groups together. So these analyses have been done in the cadenza variety, where we see this kind of profile emerging. Now, when we then zoom into the individual phosphosites, the individual peptides that we have been detecting, we kind of filter these for a localization probability above 0.75, which means that computationally and based on mass spec data, that the reliability that the phosphorylation has been assigned to a very specific residue is above 0.75. And then we do a number of additional filtering steps to really come up with those peptides that we robustly identify along the temperature gradient. And this ultimately gives us slightly over 220 phosphopeptides that are highly significantly regulated along this temperature gradient, which means that what is kind of our expectation, that within this group we should find a set of key regulators that control the processes at a given temperature. Now, one of the things that we first observed, and if you look at the temperature literature, there's kind of this distinction between what happens at cold, lower temperature, and what happens at heat or slightly higher temperature. There's always been this notion in this kind of separate research that these are two largely distinct signaling processes. However, if we overlap our data and we compare our control temperature with the either lower or higher temperatures, we do indeed see that there is a set of differential phosphorylation at each of these groups, but there is also a large overlap. And this is reflected also here if we look at the more extremes, where we do see that we get distinct phosphorylation events, but we do see about 37% of the phosphorylation events that are in common between the two different temperatures, which means that temperature signaling, if this is now cold or heat, is converging on the same molecular players. Now, in addition, what we were primarily interested in is, do we also see this kind of typical response with a maximum at 24, 29 degrees in our phosphoproteome data along this gradient? So if we cluster our phosphosites and the behavior of our phosphosites along this gradient, we do see the typical responses of upregulation and downregulation, either gradually or more kind of later, and later in this context means the higher temperature of being either up or down regulated. But we also find a number of clusters that are very similar to what we see in our profile here, which means that either in a positive way, being upregulated and peaking at 24 to 29 degrees, or kind of going down at these temperatures. So these kind of what we refer to bell-shaped clusters are reflecting the molecular players that are probably controlling these growth and developmental responses here. And you notice also here that 14 and 19 and 24 and 29 are rather similar. So this is also what we observed in the PCA plot, that indeed these two temperatures are not that different in their molecular signatures. Okay, so then the next step was to really try and identify the key components that are regulating these responses. Now, if we then look at these data in a kind of network approach, we do see this picture emerging. And the network is being generated purely based on the phosphorylation intensities along this gradient. And the approach that we use is very similar to what has been used in transcriptional networks. So if you have the transcriptional network time course, for example, the profile of the transcription factor along this time course very much matches the profile of the target genes, sometimes with a slight time delay. And by using these kind of inferences, you can connect transcription factors to their targets and build gene regulatory networks. And this is exactly what we did here for protein phosphorylation. So we did not build any kind of networks based on gene regulatory networks or based on protein-protein interactions. We purely built a network based on the phosphorylation dynamics along this gradient. And adding the assumption that if there is a phosphorylation event on a kinase, and the kinases are positioned here in the middle, if there is a phosphorylation event on those kinases, that event will be associated with an activity of that kinase. And by doing so and following the phosphorylation dynamics of the kinase over the gradient and then matching the putative substrates with similar profiles, we are able to build these networks. And this allows us to position a set of kinases that are maximally active at either the lower temperatures or maximally active at the higher temperatures. And we have a number of predicted substrates for each of these that we are currently looking into. So this allows us to indeed identify the key components that control these growth responses at each of these individual temperatures, which either have a positive role in growth promotion at the elevated, mildly elevated temperature, or a more repressive role at the colder or the extreme high temperatures. Now, in addition, and this is kind of coming back to the title of my presentation, one of the things that we aimed to achieve is to assess to what extent can we use this differential phosphorylation to improve breeding. And can we use these as protein-based biomarkers to identify specific varieties or specific genetic material that is most temperature tolerant? And can we also already identify this without having a complex experimental setup that requires growing these varieties, for example, until maturity under different conditions? So what we did is we took 4 varieties, 2 sensitive, 2 tolerant ones. We exposed these to the same kind of Q, 24 as a control, 34 as high temperature, and we do see that there are some distinct responses, with cadenza maybe responding a little bit more different compared to the other varieties. Now, most intriguingly, when we then plot the individual sites in this circus plot, what we there observe, and each of these lines is a distinct phosphopeptide, and the color coding is as such that red means upregulated, blue means downregulated upon the temperature Q, and the gray bar is essentially non-responsive. And we have the sensitive varieties here on the left and the tolerant varieties here on the right. And if you then look at where, for example, here in this sensitive variety, a responsive protein maps on the tolerant side, this very often is positioned towards the gray zone, where basically there is no response detected. And there's also kind of occurring some vice versa changes in the tolerant ones that are mapping on the non-change in the sensitive ones. And we see the same picture for both the downregulated and the upregulated ones in our data. Now, what does this actually mean? So if we zoom in to a few selected candidates, what we observe is that in our sensitive variety, we get a nice upregulation or downregulation, depending on the phosphosite that we are looking at. If we then look at our tolerant variety, where we map this to a non-change, what we do see is that it is not that there is not necessarily a change towards the up or the down regulation, comparing the control versus the treatment, but that actually under the control conditions, there is already an elevated phosphorylation status of these proteins, and therefore this does not further change. Or the level is already low under the control conditions. So the treatment does not have any additional or major additional effect, which means that under the control conditions, these tolerant varieties with respect to these peptides, phosphopeptides, are already primed, and that this could be predictive for their tolerance or sensitivity. So that also means that it is sufficient to profile the genetically encoded phosphorylation status, because this is very likely driven by a change in kinase and phosphatase activity, so that it's sufficient to quantify the phosphorylation status already under control conditions and already under, depending of course the phenotype that one is looking at, but likely already in young seedlings. And this can be predictive for tolerance and sensitivity. So this is, with respect to this part of my presentation, the status that we are currently at. So we have captured environment-regulated phosphoproteomes in sensitive and tolerant wheat varieties in the growth chamber. We are looking a little bit at a field-grown variety as well to see to what extent does this also translate outside of the growth chamber into the field. By doing this comparative analysis of the phosphoproteome data, we have been able to identify putative phosphoprotein-based biomarkers. Now, the main question that is still needs to be addressed: to what extent is this now indeed predictive for breeding? So we would like to gain access to some extent to a breeding population where there is clear physiological and genomic data to then use either phosphoproteomics or a phosphoantibody-based approach to really test, further test this hypothesis. So now I want to kind of go a little bit deeper into a few candidates. A few candidates that we have been exploring in a little bit more detail. So kind of coming back to this response of these three varieties along this gradient, there is a clear similarity in the response, although there are some differences in the amplitude. So we were wondering what are some of the proteins, apart from those kinases that I mentioned earlier, that are also potentially underlying this response. And one of the things that we observed is a specific loss of a phosphopeptide that is part of the wheat COI1. So this is the COI1 on the 1D genome that is lost in all these varieties, while the corresponding phosphoeptide on the A and B genome does not seem to be obviously changing. So this COI1 protein is potentially encoding a jasmonate receptor. The jasmonate receptor, an F-box protein that is part of an SCF complex that leads to the ubiquitination of these jas proteins, which is ultimately degrading these and facilitates a jasmonate response. So we first assessed to what extent is this COI1 protein truly encoding a functional jasmonate receptor. So in order to do that, as you noticed here, there are a number of interactions that we can test. On the one hand, the interaction with the ASK1-2 or the interaction with the jas proteins. And then we can further probe the degradation of these jas proteins. So indeed, if we take our wheat COI1 from the 1D genome and we test the interaction with the ASK proteins, we do indeed see that there is an interaction there. So on that level, this already seems to be functional, and we see that in this yeast to hybrid assay. So we also see that this ASK1 is functional because it interacts with the Arabidopsis COI1. And we also see that in this pull-down assay. So yes, on the one hand, the COI1-1D interacts with these ASK orthologs. In addition, there is also an interaction with one of these jas proteins, and we've also demonstrated that for a number of other JAS proteins, as you can see here in our yeast assay. Furthermore, when we take these wheat proteins and we put these in tobacco, so we co-express the wheat JAS9 and the wheat COI1 in tobacco, we basically see that the JAS protein disappears, which means that the presence of COI1 is able to ubiquitinate and lead to the degradation of this JAS protein. So if we specifically mutate that JAS protein to disrupt this interaction and degradation, we see that the protein stays largely stable. And we also see that if we render the COI1 non-functional by mutating that protein. So indeed, the wheat COI1 from the 1D genome is able to interact and to facilitate the degradation of these JAS proteins. Now, the final experiment that we did is to test if this wheat variant is able to complement the Arabidopsis COI1 jasmonate receptor mutant. And we did a couple of assays. One of them is using methyl jasmonate treatment, where the wild type nicely responds by a shortening of primary root growth. The receptor mutant is largely insensitive to this. Now, if we complement our Arabidopsis mutant with the wheat protein, we see that we recover the sensitivity to the jasmonate. And this is also quantified here. In addition, a typical phenotype of the Arabidopsis, or at least some of the Arabidopsis jasmonate receptor alleles, is this infertility phenotype because of the stamens not developing sufficiently. There's a lack of pollination, and this leads to poor cilique and subsequent seed development. So if we introduce the wheat protein variant in this genetic background, we can nicely recover the responsiveness of this mutant with respect to stamen growth and fertility. So further supporting that indeed the wheat COI1 1D is encoding a functional COI1 receptor. Now finally, we then also obtained wheat tilling alleles with premature stop codons in this COI1 1D variant. And indeed, when we then look at the phenotype, and here we assayed leaf sheet, leaf length, we there notice that in two independent tilling alleles, we see that compared to the control, these seedlings grow a little bit larger. And this is in agreement with some of our previous results that jasmonate is indeed repressing overall seedling growth. And this also plays a role under elevated temperature. So on the one hand, we have demonstrated that COI1 1D is encoding a functional jasmonate receptor, and that the role of this receptor is to control overall at least early seedling growth. So next, I want to kind of switch gears again a little bit and try to use the wheat data, but also some of the soybean phosphoproteome data that we have obtained to assess to what extent are there conserved temperature-regulated proteins. And these we refer to as targets of temperature, or TOT proteins. And this is what you also see reflected here in this Venn diagram comparison. If we overlay the Arabidopsis, wheat, and soybean phosphoproteomes, we have this subset of proteins that is regulated in all of these species. So these are likely functionally conserved temperature-regulated proteins. So we then selected or refined our selection a little bit further by only looking at kinases and membrane localized proteins. And there we identify one of these proteins, which we call TOT3, and this is encoding a MAP4K protein, specifically MAP4K4. And at the time when we started this analysis, not that much was known about these MAP4K proteins. So we kind of looked a little bit in the phylogenetics within the green lineage. We identified 3 distinct clades, and TOT3 is a member of this subclade 3b. So if we then look at what was known and what is at least the proposed role of these MAP4Ks in non-plant organisms, they're kind of positioned downstream of a signal, which is perceived by a receptor, which is then regulating these MAP4 kinases. And there, the name also comes from, they are positioned upstream of these canonical MAP kinase signaling cascades, MAP3Ks, 2Ks, 1Ks, which are then controlling target proteins. But there are also a number of examples where the MAP4Ks are positioned upstream of their own target proteins. And this is what we also see in plants, where in recent years these MAP4Ks have been positioned in a number of different physiological, biotic, abiotic stress growth pathways. And we're now starting to fill the gaps in this knowledge. So coming back to our selection criteria, where we were looking for membrane localized proteins, we indeed confirmed this, that TOT3 is localized to the membrane, as you can see here, with this translational fusion in GFP TOT3, where the signal is nicely overlapping with this M464 membrane dye, which kind of at least shows that it's membrane associated. We didn't see any transmembrane domain, so there's probably another component that recruits this to the plasma membrane. But the most intriguing part was, of course, when we were looking at loss of function lines, when we were looking at the phenotypes, we saw that in Arabidopsis 2 independent alleles, and we can also complement this phenotype with our TOT3 GFP fusion. We see that in wild type, we have this kind of nice growth response, this kind of lengthening of the hypocotyl, lengthening of the petioles, and this kind of upward angling of these leaves. In our 2 mutant alleles, this is also quantified here, this is largely lost. So we have much less lengthening of the hypocotyl, and this additional shoot responses are primarily gone. But what interested me most at the time when Dai, the PhD student at the time working on this, is that we did not see in these single mutants obvious effects with respect to growth, which is kind of a little bit in contrast to what has been observed in a number of other temperature-associated proteins. Now we were also, and this is also part of our filtering strategy, we were looking for conserved proteins, signaling proteins. So we were, of course, also interested to what extent do we also see weed phenotypes. And indeed, when we look at the stilling alleles, we do see that growth responses, and in this case, we measured the leaf sheet, that growth responses in these stilling alleles, and this is shown here for one example, but we see the same happening for all 3 of them, that this temperature-induced growth response is largely lost in our tilling lines. So indeed, further supporting that this response is conserved. And you can find many more details on the downstream signaling associated with this protein. But we also noticed that TOT3 is, in Arabidopsis at least, specifically expressed in guard cells. So we started to look at this in a little bit more detail, and we noticed that indeed, while we don't see any obvious changes with respect to stomata development, we do see a change with respect to the response of stomatal opening with respect to elevated temperature. So if you look at, and this is an example in Arabidopsis, our TOT3 mutant in our complementation line compared to the wild type, we do see that we get a nice response with respect to stomatal opening in the wild type and the complemented line, and this is largely gone in our mutant background. And this is also impacting stomatal conductance, so gas exchange, which we see is nicely going up in our wild type complementation line, and there is no change in our mutant background. So again, coming back to the question, is this now conserved? So we went to our tilling alleles for TOT3, and we indeed see that while the wild type is nicely responding both with respect to the stomatal opening and with respect to stomatal conductance, the mutant no longer shows any kind of response. So we then started to look into a little bit more detail what would be underlying these responses. And just to make a very long story short, because this has been several years of work that I will now summarize in a few slides by mainly Xiangyun Xu within the group during his PhD, we noticed that TOT3 gets activated by heat, and we see this in this autophosphorylation quantification of this autophosphorylation, and that this active TOT3 is directly phosphorylating the proton pump. And this is reflected here, that TOT3 leads to an in vitro kinase assay phosphorylation of the proton pump. And if we then mutate the phosphorylated residue, which is associated with controlling the activity of the proton pump, we lose this phosphorylation. And the proton pump controls of course, the movement of or the establishment of a proton gradient and the movement of these protons, which is also activated by heat, which is not occurring in the TOT3 mutant. So we have an activation of the proton pump, which leads to stomatal opening, and this leads to leaf cooling. Now, as I mentioned, high temperature is very often coinciding with drought conditions, but under drought conditions, you want to close your stomata and retain water. So under these combined stress conditions, there is obviously a conflict. So what we noticed is that under drought conditions, ABA is produced, and this leads to the activation of OST1, one of these ABA signaling components, a SNRK kinase, which leads to stomatal closure. We also noticed that under drought or ABA conditions, as you can see here quantified again, quantification of these autoradiograms, we see that there is less active TOT3 through ABA or under drought conditions. So this means that TOT3 gets inactivated, and it gets inactivated through differential phosphorylation by this OST1 SNRK kinase. So we could show transiently in tobacco that these residues are being phosphorylated in tobacco in the presence of ABA and OST1 on the TOT3 protein, and that indeed this happens directly because, again, in vitro we see phosphorylation of this TOT3 fragment, and if we then mutate these residues, we lose the phosphorylation. So there's a direct phosphorylation of OST1, and this leads to the inactivation of the protein. Because if we mutate these sites so they can no longer be phosphorylated, we have a less active TOT3 compared if you mutate these sites, we have a more active TOT3 compared to the control, as shown here with respect to autophosphorylation and the proton pump target. If we then mutate these into a phosphomimetic, we see less activity with respect to autophosphorylation and the target protein. And this is also reflected in the complementation potential. So if we have a phosphomutant, which can no longer be phosphorylated and which is more active, this nicely complements the stomatal phenotype. If we have a phosphomimetic, we see that this is a less active protein, and this can no longer complement our TOT3 mutant. And we see this also reflected in the proton pump activity. So we see elevated activity and elevated proton pump activity in our alanine variant, showing that this is indeed a hyperactive one. And we also see that this phosphomutant variant is less sensitive to ABA-induced stomatal closure, as you can see here. And indeed, under drought conditions or ABA conditions, at least one of these residues is more phosphorylated in the presence of ABA or drought in an OST-dependent manner. So this kind of further supports that indeed also in seedlings, when they are perceiving drought, the regulation of this residue is essential to control the activity of the TOT3 protein. So this kind of leads to the following model that under high temperature, TOT3 activates the proton pump, which leads to stomatal opening and plant cooling. However, when this is coinciding with drought, and this also very much depends on the intensity of the drought signal, there's a production of ABA, which activates the OST1 SNRK kinase, which leads to phosphorylation of TOT3 and subsequent inactivation of the TOT3 protein, which shuts down this particular pathway and allows stomatal closure and then retaining water instead of losing water through open stomata. Okay, so with that, I want to just kind of give you a couple of take-home messages. So I hope that I was able to show you that capturing protein phosphorylation allows the identification of, on the one hand, novel signaling pathways, including components that control temperature signaling and plasticity along a temperature gradient, and that these phosphorylated proteins can be valuable breeding markers, protein-based breeding markers, that using conserved phosphorylation events allows the identification of conserved regulators, such as TOT3, which is conserved in its function in wheat and Arabidopsis, and that the TOT3 AHA1 proton pump OST1 signaling module integrates different environmental signaling pathways, in this case heat and drought, and that COI11D from wheat encodes a jasmonate receptor that is indeed involved in regulating wheat growth. So with that, I just want to close with thanking the people who did the actual work. So what I presented with respect to the wheat analysis is mainly the work of Cassio during his PhD with the help of Dai, and also wheat-related work done by Ting Ting. The OST1 TOT3 work is mainly the work of Xiang Yu in his PhD with the help and guidance of Dai, and additional recent experimental support from Hong Yang. And then, of course, with the help of our technician, Brigitte, a number of collaborators that contributed to this work. So with that, I want to thank you for your attention, and I'm happy to take any questions.
Wonderful. That was a really, really interesting seminar. Thank you so much. There are just a few questions. The first is, how many of these potential protein phosphorylation breeding markers have you identified, and are they associated with allelic variation in the target protein?
So we initially kind of identified or selected a small set of about 10, which of course we still need to further validate. We then did try to match these using the wheat genome variety data to see if indeed there are certain positions that might also be selected for within different temperature tolerant, temperature sensitive varieties. Using a very kind of crude approach and just looking at the phosphosites, we did not really pick anything for the candidates that we identified. However, it might be a little bit more complex. The variation might lie within a larger region that then prevents docking of the kinase and phosphatase to control this, but it might also lie on the level of the regulatory kinase and phosphatase, which is something that, because we don't have the information, or at least the confirmed information on what regulates these, which is something that we have not yet been able to explore at the moment.
Okay, and kind of maybe a related question is whether TOT3 is associated with any of the known heat tolerance QTLs in wheat. Has that been?
We haven't checked that. So that I think that's something that we should explore at some point. So we've looked a little bit at the known heat signaling components in Arabidopsis, and there it seems to be largely independent of the known hormonal phytochrome associated signaling pathways. We have not looked to what extent it also overlaps with wheat QTLs, etc. That's a pretty good idea that we need to look into.
Okay. And here's a question. What are your suggested other PTM mechanisms for drought? What is a possible mechanism if combined stress, heat plus drought, versus individual stress is imposed?
So what we're kind of looking at is signal integration. So I think it's this balance of protein phosphorylation on different residues, which kind of is as a whole making up the phosphocoat. And it's this kind of phosphocoat that will determine the output of this particular protein. So if the phosphorylation goes 100% towards, in this case, drought-mediated phosphorylation and inactivation of the protein, this protein will not be active. Of course, if there is only a mild stress, there might only be a fraction being phosphorylated and inactivated. So it is really this, it's this gradient, this substoichiometric gradient of phosphorylated proteins, dot proteins in this case, that will determine the output. There's probably also a tissue, cell type, organ type specific regulation that plays a role, and of course other modifications impinging on these proteins will further control the activity of these proteins as well.
Okay. There's a kind of I guess, a methodological question. So how is the phospho enrichment performed?
Okay, so what we do, and I just kind of very briefly alluded to this, is we take the proteome, we chop these up in small pieces using trypsin, so then you have little fragments, and then within these fragments are the phosphorylated residues, which we then enrich using titanium IMEC beads. So we have beads that recognize the phosphorylation on these peptides. So we can attach these to the beads, and then we isolate the beads, and we isolate then the fragments from these beads, and then we subject these to mass spectrometry for quantification and identification.
Okay. All right, maybe there's time for just one more question. One of them is kind of complicated. Maybe you could answer offline. The other one is, are there other candidates found in phosphoproteomics? How do you pick COI and TOT3 out of the candidates?
Yeah, so the top three we basically picked because we had a number of filtering steps based on our interest: plasma membrane localization, kinase activity, and that's something that is kind of the one candidate that we pick based on that. Within that larger set of dot proteins, targets of temperature, there are a number of other ones as well. So there's, for example, SLOMO, which is an FBOX protein, where we have been looking into its role predominantly in Arabidopsis because we struggled a little bit with the wheat data. And there are a number of other putative targets as well. For COI1, we basically went for the very specific phosphorylation profile because there we really saw that we lost the phosphopeptide completely upon elevated temperature. So we were really kind of curious to see what was happening there. And we were kind of more or less able to determine that it's a loss of phosphorylation and not a loss of the overall protein. So we're now kind of trying to build a story where this phosphorylation event itself is being mechanistically, functionally characterized, and what's the role of the COI1 phosphorylation.
Okay, well, thank you so much. It was really interesting, and you're
Thanks a
definitely
lot.
taking pioneering approaches and the combination of using molecular genetics in Arabidopsis and in wheat and phosphoprotein identification and network analysis. It's really fantastic work. We are so appreciative that you were able to take this time. And I also want to thank everybody who's online who attended. We really appreciate the participation and the community that we're able to generate using these webinars. So thank you, everybody, and thank you, Eve, again. And I wish all of you a terrific day.
Thank you. Bye bye.
Thank you so much.